A medical technician can take a prepared sample of blood and determine the concentration of hemoglobin present in the blood. This determination is done by a technique called spectrophotometric analysis. Spectrophotometric analysis uses the principles of Beer's law. Beer's law was formulated about 200 years ago. However, the foundation upon which Beer's law is based is rooted in man's basic understanding of the nature of light. In 1666, Isaac Newton demonstrated that white light was composed of all the colors visible to the eye. In his small laboratory, Newton placed a prism in a beam of sunlight streaming in through his window. The sunlight refracted by the prism revealed a range of colors when the light struck a white surface. Newton further discovered that if he used a lens to reconverge this refracted light, it would again appear as white light. Newton's discoveries were revolutionary. He was able to prove that sunlight is comprised of all the colors visible to the eye. However, this visible light, in fact, occupies only a small portion of the entire range of radiant energy, which is known as the electromagnetic spectrum. As Newton discovered, the visible portion of the electromagnetic spectrum is divided into wavelengths that are detected by the human eye as different colors. It is the interaction of radiation of these wavelengths with an object that causes the object to have a color. When light hits an object, specific wavelengths of the electromagnetic radiation are absorbed, and the rest of the light is reflected back to the eye. The reflected light, that is, the visible light minus those wavelengths absorbed by the object, is detected by the eye as the color of the object. If the absorbed light could be seen, it would be blue, the complementary color of orange. To better understand complementary colors, consider this simple demonstration. An orange appears normal under white light. If the color of the light is changed to orange, the object still has an orange color. But under blue light, the complementary color of orange, the fruit appears black. No color is being reflected in this case because all the wavelengths of the blue light are being absorbed. Besides blue and orange, other pairs of complementary colors are red and green, purple and yellow. These are often depicted by a color wheel. Another example of complementary colors can be seen in a demonstration. The equipment consists of a source that generates a light beam, a holder that allows a colored solution to be placed in the light beam or out of the light beam, a prism that disperses the light into its component colors, a lens that can recombine the dispersed light, and a screen on which the light is projected. Before the colored solution is pushed into the holder, the light beam shines directly on the prism, and the familiar spectrum of red, orange, yellow, green, blue, and violet light results. When the blue solution is pushed partially into the path of the light beam, some of the light passes through the solution, and some continues to shine directly on the prism. Due to the optics of the equipment, the light passing through the solution and prism is seen on the bottom of the TV monitor you are viewing. Because orange and blue are complementary colors, the orange wavelengths are being absorbed by the blue solution, and thus removed from the spectrum. When the solution is pushed fully into the holder and a lens is placed in this beam of light from which the orange wavelengths have been removed, the recombined light has the same blue color as the original solution. This is because the visible light without the orange wavelengths that were absorbed by the solution is blue. In order to determine the amount of the absorbing species in this blue solution, it is necessary to study the ability of the solution to absorb orange light, its complementary color.
The instrument to do this is called a spectrophotometer. It includes the components of the apparatus just seen. Although most spectrophotometers have opaque covers that are sealed, this one has been designed to illustrate the internal operation of the instrument. The basic spectrophotometer combines in one unit a spectrometer and a photometer. The spectrometer consists of a light source, a wavelength control knob, a prism or grating, and a slit. In this case, the light source is a bulb which provides white light. The light is focused onto a mirror and then reflected onto the grating. The grating disperses the light into a spectrum of colors. By turning the wavelength control, the angle of the mirror is changed and the position of the spectrum that falls on the slit is moved. The narrowness of the slit allows only a small portion of the spectrum to pass through the solution in the cell on the other side of the slit. The photometer portion of the spectrophotometer consists of a phototube which detects the transmitted light and changes it to a weak electrical signal. The signal is amplified and displayed on a meter, usually marked in both the units of absorbance and percent transmittance. When any light is absorbed by the solution, less is transmitted through the solution and detected by the phototube. That is, as absorbance increases, percent transmittance decreases. By adjusting the wavelengths of light allowed to pass through the slit and the solution, the amount of light absorbed by the solution at each wavelength can be determined. In each solution there is a specific wavelength at which the absorbance is a maximum. Besides the wavelength, two other factors determine the amount of light absorbed by a particular solution. These are the concentration of the solution and the length of the light path through the solution. In this example, one blue copper amine complex solution is more concentrated than the other. Since the tubes are the same width, the length of light path, often called the cell length, is the same. Remember that the light passes through only a small section of the solution directly behind the slit. The cell length is the distance the light travels through the solution. Between 1789 and 1852, several noted scientists were involved in formulating a mathematical relationship between absorbance concentration and cell length. The resulting equation was one of the first mathematical expressions of a scientific phenomenon. Beer's law states that the absorbance A at a given wavelength is directly proportional to the cell length, B, and the concentration, C. If the concentration is measured in moles per liter and the cell length is in centimeters, then the proportionality constant is called the molar absorptivity and is represented by the Greek symbol epsilon. Beer's law is therefore commonly written as A equals epsilon B C. According to this equation, a plot of absorbance versus concentration should give a straight line that passes through the origin. Once the plot has been obtained, it can be used to determine the concentration of an unknown solution. This is the most common application of Beer's law. In order to obtain the absorbance of the solutions necessary to plot this graph, a spectrophotometer is used. However, before any measurements can be taken, the limits of the meter on the spectrophotometer must be set. To set these, a solution known as a blank is prepared. This blank solution is often distilled water, but it need not be. When the unknown solution contains many species besides the one whose concentration is being determined, the blank contains everything except the unknown species. The glass sample tube, called the cuvette, is filled with the blank solution, wiped with a soft, dry, lint-free tissue, and inserted into the spectrophotometer with the marks on the tube and the spectrophotometer aligned. The control knobs are adjusted to set the meter to read 100% transmittance or zero absorbance when the light beam passes through the blank solution, and zero transmittance or infinite absorbance when there is no light passing through the solution. When these calibration adjustments are completed, the spectrophotometer is ready for measurement of colored solutions, such as this blue copper amine complex solution. There are 3 steps in the procedure to analyze a colored solution. The first step is to obtain a spectrum or graph of the amount of light absorbed versus the wavelength of light. The colored solution is placed in the spectrophotometer, and the wavelength is changed systematically to obtain measurements of wavelengths and the corresponding absorbance. These data are recorded and plotted. The wavelength, usually measured in nanometers, is plotted against the absorbance to give the spectrum of the solution. The wavelength that corresponds to the greatest absorbance is usually selected for the subsequent Beer's law measurements. If the solution is too concentrated, the maximum absorbance of the spectrum will be infinite over a range of wavelengths. In these cases, the solution must be diluted in order to obtain the maximum absorbance for the second step of the procedure. The second step in the analysis is to obtain the Beer's law plot of the absorbance versus concentration. several solutions of known concentration as well as the blank are needed. Before this second step, the wavelength is set at the value selected from the graph of the spectrum. It is a good practice to check that the limits of the meter have not changed. The blank is inserted and the zero absorbance is checked. The infinite absorbance is checked with no light passing through the spectrophotometer. After this check, the cuvette is rinsed with the most dilute standard solution. After several rinsings, the cuvette is filled with the solution and wiped dry with a lint-free tissue and then placed in the spectrophotometer. Absorbance and percent transmittance are read and recorded in the notebook. Both measurements should be noted and recorded. Since absorbance and percent transmittance are related to each other mathematically, they can each be calculated from the other. In this way, the second reading can be used as an experimental check for the accuracy of the first reading. These measurements are repeated for the standard solutions of known concentrations in order of increasing concentration. Once the data have been obtained, absorbance and concentration can be plotted and the best line determined according to Beer's law. This is a calibration line for a particular system. Beer's law applies only to dilute solutions. Sometimes the relationship between absorbance and concentration is not a straight line at high concentrations. Usually, only the straight line portion of the calibration line is used for subsequent analysis of unknown solutions. The third step in the analysis is to measure the absorbance of the solution of unknown concentration. From the slope of the calibration line and the absorbance of the unknown solution, the concentration of the unknown solution can easily be calculated. Because a measurement is involved, there is some error involved in the determined concentration. The manufacturer can give the reproducibility of the absorbance measurements on the spectrophotometer. This is often of the order of 1 or 2% transmittance. By calculating this error in terms of absorbance, a range of possible absorbances for that measurement can be obtained. In addition, the errors in the solutions of known concentration can be determined from the tolerances of the equipment used to produce these solutions. If these errors are known, or can be determined, the uncertainty in the value of the concentration of the unknown determined from the Beer's law plot can be estimated.
This type of analysis has many practical applications, from the monitoring of the lead concentration in gasoline to the evaluation of the grade of tomato paste. The health sciences and research laboratories also depend extensively on spectrophotometric analysis as a fundamental analytical technique.